Well, thank you for joining us. I'm Alison Moran at Royal Arts University, and today what we're going to do is we're going to look at a little bit of molecular biology in the lab. Now, environmental science has become much broader over the last few years, and molecular biology is a tool that gets used quite a lot now. And so our students need to learn basic techniques that are used, and so one of the typical techniques is what we're going to do today. We're going to actually isolate plasmid, which is a small self-replicating piece of DNA within a cell. Okay. And so we'll go on and talk a lot more about this as we go through, but why don't we just dive in today?
Great.
So Philippa, if you and Sharon would please put 1 ml of culture into the tube on the left. First thing you have to do is adjust your pipettes to 1000, which is 1 milliliter. And we're going to take some cells that I've grown up overnight. And these are E. coli that have an antibiotic resistance on the plasmid that we're going to isolate. So I grew up the cells in the presence of the antibiotic so that all the cells would have plasmid in it, so we can actually isolate.
And pop it in here?
Yes, please.
Great.
Once we have 1 ml of culture, and if you discard your tip in the tip discard, please.
Yes.
There we go. Put your lid back on, and let's give these cells a bit of a spin so we bring them down to the bottom of the tube.
Great.
So what we're doing now is we're basically taking an overnight culture and we're spinning the cells down to the bottom of the tube so that we can actually extract the DNA from those cells. So if you just hold the button down for a while, let's give it a little bit. Let's wait for it to rev up. And sometimes we have to wait for these things to rev up. Now it's fast. Give it about a count of 5.
1, 2, 3, 4, 5.
Okay, that'll probably be good enough. Now what you're going to do is remove what we call the supinate. So let's have a look at what you've done now. You've spun that out.
Okay.
Okay, so let's lift our lid and take cells out. There we go. And now let's have a look at it. Can you see how we have very clear broth now with our cell pellet at the base?
Yeah.
So what I want you to do is I want you to remove this broth. So just use your pipette again, remove the broth, and then what we will do is resuspend the cells in the first solution.
Okay.
So first thing to do is take the broth off the top, the supinate. And that will go into the liquid discard. You have to go very straight down on that.
Okay.
And so push the air out. There you go. And suck up, staying away from your pellet. But try and remove as much of the broth as possible. Just keep pushing your tip down into the broth. There you go. There you go. Keep going. Keep going. Down, down, down. A little bit more. There you go. Okay, I'd actually just get rid of that and then suck that last little bit of broth off. There you go, if you can. Yeah. Sometimes it just needs an extra suck. There we go. Okay. Good stuff. So now the first thing we need to do before we can, yes, it's drop your pen. The first thing we need to do before we can actually extract our plasma is we need to resuspend that pellet. So if you pop your pellet down and readjust your pipette to 250 microliters. So if 1 ml is 1,000th of that liter of milk that you have, we're now going down to 1/4 of what we had before, 250 microliters. And once you're at that, if you just lock your pipette, we can then have you take 250 microliters of a fluid for resuspension.
Okay.
So what we're looking to do here is just get the pellet back up in solution. So here we go, 250 microliters. Nice technique, nice and straight. Good. And Jan, if you'd like to do that. And then pipette up and down to resuspend your pellet. So
pop this in.
Pop it in and then pipette up and down. We call that trituration. Gently up and down to resuspend your pellet. That's good. That looks well resuspended. That's enough. Yeah.
Great.
That was very vigorous. Okay, so let's pop that down. Now, buffer 2 is going to do the work. Well, now what we need to do is we need to blow the cell apart. And we're going to do something called alkaline lysis. So we're adding alkali in this.
Okay.
And that is going to solubilize, it's going to unwind the proteins and unwind the chromosomal DNA. It won't really do much to our little plasmids, but we're unwinding everything else. So if you gently add 250 microliters of this buffer to your cells, be very upright. I think that's maybe needing it. There we go.
Bit of a push.
Okay, you're good. Gently suck up. There we go. And then add that to your cells. Now, what I would like you to do is hand me your pipette. Now, first off, we had cells that were kind of cloudy, didn't we?
Yes.
Now, I would like you to shut the lid and flip it up and down a little bit. There we go. And it's kind of taking on a blue tinge. And that blue tinge is to tell us that we've got the alkali in there. Okay, now as we're looking at it, let's put it up to the light. I think you're going to need to go back and forth a little bit more on vigorousness, actually. Yeah, really give it a shake. There we go. Yes. And now we're getting there, but you can see how it's gone clear.
Yeah.
So instead of having those cloudy cells there, you've blown them apart. Now I'd like to show you something about your cells. If we look at this, you can see it gets a little stringy. You see these little strings? That's your chromosomal DNA. Because it's quite long and large, it will actually unwind, and all the cells' long chromosomes have unwound and tangled together, effectively creating a string. So it becomes very viscous. Now, we've unwound, okay, we've got everything in solution now. We've unwound our proteins and we have unwound our chromosomal DNA, but we want to separate them away from the plasmid. So how are we going to do that? Well, we now take it from alkali and we're going to neutralize the solution. And what's going to happen is those proteins in that chromosome are going to wind back up, but because they're doing so quickly, they end up all clumped together. So we're basically going to precipitate them so that they precipitate away from the nice plasmid that we're hoping to isolate today. So here we are. Let's have you just at the 350 this time. And we are going to add 350 microliters of the neutralization buffer. There we go. So, you can add that, please. All right. And we know we're neutralizing because our blue, if you shut the lid and start flipping it back and forth, you can see the blue color go and we're precipitating out. You can see how all those proteins and chromosomal material have started to precipitate out as this white gunk.
Yeah.
And our plasmid, that small self-replicating piece of DNA, is going to stay in solution. So that's going to be what we want to do next. So if you're both ready, What we would normally do is spin this for 15 minutes to get rid of the plasmid.
Okay.
Having said that, because we didn't want to spend 15 minutes watching the spinner, we prepared one earlier.
Great.
So this is a Julia Child moment. So let's pop them in the spinners, but take out the ones that we prepared earlier. And something you'll notice is you're always putting them in equal and opposite, and that's because such rapid rotation of the spinner, if they were imbalanced, would break the spinner.
Okay.
Because you're creating such large forces. And believe me, it'll really grind if you have it imbalanced. So that's why you're being very careful about your position. Okay, so now the next thing to do is to take the clear supernatant where you have your plasmid and separate it from all the gunk that we've precipitated. And I want you to put your plasmid into this spin tube. So here I have a tube with a filter, and that filter sits in a collection. This collection tube is just going to collect the waste material, the waste flow through.
Okay.
Now, this is something that we can do without a kit. It's just typically in labs and research labs and so on, people use kits because they're quick and convenient, and so we want our students to basically have a chance using a lot of the typical techniques that'll be used in a lab. So that's
Sure.
why we're giving this experience is using a kit. This is a very common kit. It's very it's good quality. You get a good product from it. So
Great.
that's why the students are using it. So if you would add your clear supernatant to this, you might want to put your tube down for a second.
Sure.
And adjust to maybe about a mil, just so you've got more than enough room. And then you can transfer the fluid into that tube.
So I open this one up.
Just open it up, yes, and try and stay away from the white.
Sure.
The important stuff is to have the clear fluid with as little white as possible. Now, unfortunately it's slumped a little bit to the base, but that'll be fine. It's better to take less fluid and stay away from the white. So just suck up. It should be quite good. There you go. You're doing a very good job for a beginner pipetter. You've got enough there. That's more than enough.
Great.
Good. And then just add it to your loose spin tube. Good. Now the idea behind this spin tube is that we get rid of the liquid and a lot of extra, say, carbohydrates and so on that might be around. And you can that's all waste, so we can throw that away too.
Throw this in here.
Into the tip discard.
Into the
There
tip
we go.
discard. Great.
But that filter, our DNA is actually going to bind to that filter, so we'll be able to not only concentrate it, but wash it and clean it
Okay.
with solvents. So why don't you and Sharon pop your spin tubes into the spinner oppositely and give it a quick spin. Now you don't need to put the lid on this one.
So I can just close
Yeah,
this.
just close that.
And how long should I spin it for?
Just get
Give
it
it a-
going for
get
a
it
bit,
going?
because
Sure.
it'll take long. I don't know time. That'll be good. I don't know. Sometimes it's just sort of a sound. Oh no, that'll be done. Okay.
Great.
So now when you get that out, you're going to discard that flow through. Remember, we were concentrating our DNA on the filter. So you can discard your flow through in the liquid discard. And then put your...
So this here into
just
the liquid.
discard the liquid.
Yes.
And then put your filter back into the collection tube. Now the next thing we need to do is we need to wash it with some solvent. So let's pop it back down and adjust this to 750, and we're going to add 750 microliters worth as a solvent. So we're washing our DNA, we're getting it clean, we're keeping it nice and precipitated, and we're going to actually be able to get a nice clean product now. So if you gently suck up. Good. Now add that onto the filter. There we go. Onto the tube. There you go. And again, you and Sharon are going to do the same thing again. You're going to spin that through.
Okay.
So we're basically just washing our DNA through with the solvent. So if you just spin it as you did last time, that would be perfect. It's a little noisier without the extra lid on it, isn't it? Yeah. Okay, so let's discard our supernatant. And one thing I like to do at this stage is to give my tube an extra spin because let's discard our supernatant into our liquid discard. But you'll often find that there is a little bit of solvent still on the sides of the tube, and to make sure that there's no solvent still around, we give it one extra little spin, and that just makes sure that it's taken it away, because if the solvent's there, we won't be able to resuspend our DNA. It'll keep it in the precipitated state.
Okay.
So give it one last little quick
spin.
whiz in the spinner, and then we can move on.
Great.
Right, and at this point, we are now ready to resuspend our DNA that you've prepared. So let's just pop this down for a second and think about what we're going to do. What we're going to do is move your blue spin tube into a fresh tube, and we're going to add a resuspension buffer that's at pH 8. pH 8 is a pH that DNA really likes to come into solution in. So what we're going to do is we'll add that pH 8 buffer to the filter. We'll let it sit for a little bit, and then we'll spin it through and we'll have prepared DNA.
Great.
Okay. So this is actually set at 50, so that's great.
Okay.
And so you can move your blue tube across to your clean Eppendorf. Eppendorf's just the name for these 1.5 ml tubes that we use regularly in microbiology.
Okay, so if I open this up and just pop the blue, let me pop that down.
Yeah, pop the blue filter in.
Okay.
There you go.
In that.
There you go.
Great, thank you.
And throw away that. That's excellent.
Great.
Okay. So now what we're going to do is you're going to take 50 microliters, and what I want you to do here is I don't want you to touch the filter, but I want you to try and add it near the center of the filter. Just slowly dispense it so it really is hitting that filter.
Okay.
There you go. Now, as you know, we're going down in volume here. We're using yellow tips, and so the yellow tips are for the smaller volumes, and this time we're using 50 microliters. So we're down again. If 1 microliter is a millionth of that liter of milk, we're using 50 millionths of that liter of milk.
Wow.
I know, it's a pretty small volume, but we're going to go smaller yet.
Okay.
It's the how small can we go challenge day.
Great.
Oh, you're doing a very good job. You would be most welcome in our class, that's for sure. Okay, so now let's pop it down and think about what we've done so far. So to reprise, we were trying to isolate a small self-replicating circular piece of DNA.
Yes.
So that's separate from the chromosome. And so what we did was we spun down cells containing that plasmid. We then resuspended them and blew them apart. We did cell lysis. That lysis means to break.
Yes.
We lysed the cells. And we lysed them in alkaline. So everything unwound. And the big chromosomes all unwound, and they all got tangled together and became quite stringy, and all the proteins unwound. And then we very rapidly brought the pH back to neutral, so with some acetate. So what happened was they all precipitated because they tried to wind up too quickly. And that created that white clumping because they just randomly came out of solution because they all unwound just in a disorganized manner.
Yeah.
And so we were able to spin those away, and all that was remaining in the fluid were things that we were wanting. We got rid of the chromosomal DNA and the proteins. We then ran that supernatant across a filter to which the DNA bound. We washed it through, and now what we've done is we've added a buffer the DNA will resuspend into, and we'll be able to have our DNA and use it. So
great.
Let's spin this down and see what our final DNA looks like. There you go. Something else. Oh well, it's well.
It's had a good spin.
It's really
good.
Spin. There you go. Okay. So let's pull our samples out.
Sorry.
And if you look in the bottom of the tube, you now have your DNA sample. It's a lot smaller than before, but it's nice and clean and clear, isn't it? So quite a difference from that murky broth that we started with.
Yes.
Okay?
That's super clear.
So now we can get rid of that spin tube, and we're going to consider how we look at DNA. So let's just put our lid on for a moment.
Okay.
There we go. There you go. Now let's pop it down and talk about how molecular biologists often look at DNA. So something that they tend to do is they tend to run DNA in gels. And that helps us decide how big it is. So here's a typical gel kit, and I've just cut a little bit of gel out. And you see there's these wells, these little gaps. We can load our DNA into these wells and then run it under an electrical current to separate it by size. Now DNA is negatively charged, and so when you put it in an electrical current, it will move to the opposite pole. Large fragments of DNA have more trouble navigating through the matrix of the gel, and so they are slower than tiny little pieces. And so what you'll end up getting is a nice separation of sizes. And to guesstimate how big your pieces are, we often run a molecular weight ladder along with our gels so we can see exactly how far they've run.
Okay.
So we're going to actually look at how you load a gel right now, and then we'll talk about what we did and see the dye front move through.
Great.
Now here we've got a gel loading dye, and you can see it's nice and blue, and it also contains glycerol. And so I'm going to ask Sharon to put out some 1 microliter blobs of gel loading dye. Now what we tend to do in molecular biology is load lots of samples at once. So in a gel I may have 20 samples or so on. So I'll tend to just run a number of little dots of my gel loading dye along on a parafilm or a piece of saran wrap, and then add samples and mix them and then load them to the gel. So Sharon's going to load our first gel, and she's adding this loading dye to the parafilm. And what you're going to do is you're going to take some of your sample, mix it to the loading dye, and then put it into that well. And the glycerol is dense, so it makes the sample sink into the well. And there she goes. Okay, so maybe Sharon, if you would load 5 microliters of your DNA sample, just to show Philippa how it goes. And then we'll let you have a go, Philippa, and you can load some of your sample onto it. No, you're going to have to use the other.
Okay.
We're really going small.
Small, yes.
So now she was putting 1 microliter dots down there, and she's going to load 5 microliters. So 1 microliter is that millionth of that liter of milk that you buy in the store. It's pretty small.
Tiny.
And so she's going to mix it up and down. She's going to triturate the sample. And suck it up. And what I find really helps with this sort of thing is balancing yourself. And
yes.
So everybody's got their own way of balancing. Doesn't really matter. But balancing yourself so you're nice and steady, and then you can just load it into the gel. And you can see that it's sinking into the well. Let's try another one here. I'll do one just for fun. So here's 5 microliters, up and down. And I'll load it into the gel. So I actually put the tip into the gel, and for some reason it's flipping out. I do not know why.
Okay.
That's unusual. Yes, now I see why you had a problem. Didn't do that earlier. Okay, let's give you a, okay. Just do your sample. Let's see how it goes.
Sure. Okay.
Okay, you're good.
Okay.
On you go. Okay.
Great.
So, kind of went a little bit funny this time. Never quite had it go funny before, but there we go.
Well.
It was the first time for everything. Something didn't quite work out. So I'm actually just going to load some dye in so that we can actually see some dye front move. Okay, I'm not quite sure what happened there, but there we go. Alright, so what we want to do is watch the dye front move. And in general, we use this blue dye because it tells us how far things have gone. And it gives us an indicator so we don't run our DNA off the end of the gel. So what I've done here is I've got the gel running, and it's going to start to pick up and voltage and then run at a constant current. So if I look down here at the electrodes, see the wire going along, you can see the bubbles rising up, and I can see the current is now flowing between the 2 electrodes. So what I'll do is I'll wait until it's moved through, but we can have a little chat about what we're doing and then come back and see it moving through that gel. Okay,
Okay, great.
All right, so let's just use the drawing board for a second and think about what we're doing here. So if I imagine my cell, an E. coli sort of rectangular shape, so we'll go with the rectangle. We have our chromosome, which is big and large, and that's where all the code for and the instructions for the cell are. Okay?
Yep.
So all the instructions are there, but we've been talking about something called a plasmid, which is a small self-replicating piece of DNA. And this plasmid has something called antibiotic resistance on it. Now, you've probably heard your doctor say, the thing about not overusing antibiotics and so on.
Yes.
But what a lot of people don't realize is that antibiotics are normal, common products in the soil. The soil is a chemical war zone, and all the different organisms are competing against each other, and part of the armaments are antibiotics. So if you look at most of our common antibiotics, they are from soil organisms. So they're naturally there, and we've tried to use them for our benefit. But in warfare, in the arms race, what do you do if you have a weapon? You make something against the weapon. And so antibiotic resistance is another way that organisms compete. So if a competitor has an antibiotic, you have an antibiotic resistance against it. When we misuse antibiotics, we basically make sure that only organisms that have resistance can grow. And so it's very important that we don't. But in molecular biology, we've made use of one of the things that this antibiotic resistance. So a natural place that bacteria might have an antibiotic resistance is on one of these plasmids. And they're very, very caring and sharing, bacteria are. And they'll donate these plasmids to each other. And so that's one of the reasons why it can be a problem in human health. But in molecular biology, it can be really useful for things like doing manipulations. And of course, molecular biology is used from everything from determining community structure to developing methods for bioremediation and so on. So it's really a technique-based discipline.
Yeah.
And so these plasmids are very useful. And something we often do with these plasmids, which is what we've just isolated, now I'm going to make a circle. Something we do is we use them to carry types of genetic components, codes that we want them to do. And we can select for them with this resistance. So something I can do is I can cut it with something called a restriction enzyme. So a restriction enzyme is an enzyme that cuts DNA.
Okay.
And bacteria tend to make these restriction enzymes as part of their competition again. You have an immune system, you make antibodies against nasties that may come into your body, anything non-self, and they would attack it. Much the same way, bacteria have restriction enzymes that recognize sections of code that are not the same as themselves.
Okay.
So non-self code. So they recognize words they don't have. And when you look at this name, expa, it actually stands for the organism that it came from. It's a type of Xanthomonas. So say I've cut my plasmid here, and say my plasmid is almost 2.7 kilobases, 1,000 bases, roughly. And say I use a second enzyme here and cut that. You can see I'm going to get two unequal strands. Okay. So let's take my little piece of tape here.
Yes.
And I've got my circular plasmid. Now what happens if I cut my plasmid once?
You just have one long
I have
strand.
one long strand. Okay. What happens if I cut my plasmid twice?
You have two separate strands.
Okay, so I get a long and a short strand.
Yes.
I can now cut it and run it under electrophoresis and see my strands. So
Okay.
let's look at how we might image this DNA, okay?
Okay, great.
So how's our DNA doing in here? It's moved, well, our buffer has moved a little bit. So let's just look at that. What I want you to see is that you can see how the blues moved into the gel. And
Yes.
so that's it moving into the gel under the electrical gradient, okay?
Okay.
So now let's look at what it actually looks like if I should actually happen to cut. Have a look in there. Think you need to be up a little bit? There you go. Can you see that we actually have a ladder on the left and then a single band that's a little bit lower than the one next to it?
Yeah.
And then two pieces. So I've got actually a picture here where you can look at what we're looking at. So this is this molecular ladder I was talking about, and here is a band at 1 kilobase. This big band here, this is the uncut plasmid, and because it's all tightly wound, it can actually wiggle through the gel a little bit faster.
Okay.
Here we've cut with one enzyme. So if we looked, we'd say, oh, that's just about 2.7 kilobases. Here, I've cut it with two enzymes. And see, I've got a large piece and a small piece. That one's sort of about 1.9 and that one's about 800. So add them up and you get your full plasmid. So you can see how I can see the DNA on the gel. And now you'll notice it kind of looks green, doesn't it?
Yes.
So what we've done is we've added a dye that binds to the DNA.
Okay.
And when we shine UV light through it, it absorbs that UV light and then it fluoresces in the green. So fluorescence is the absorption of energy at a higher wavelength and then at like UV, and then putting it out at a lower energy level, which is in the visible spectrum, the green, and that's why you're seeing your DNA as green on that gel.
Okay.
Okay, so here you can see how imaging of your DNA can occur. So in molecular biology, we all often prepare plasmids. We are often checking fragment size. These are very, very typical components of your everyday job if you're working in the lab. So thank you very much.
Thank you. That was fascinating.
Everything.
Yes.
And you did very, very well.
Thank you.
Isolating your DNA.
Thank you. Love it. Thank you.
Okay. Bye-bye. Yeah, so you can really see the ladders there and the single band, and see how it jumps up because it's all twisted up so it can wiggle through a little bit faster there, and then the separation of the two bands. And if I'd run this further, you'd see it separate out even a little bit more.
Fascinating.
Yeah.
That's
and why don't you have
great.
another look at what it looks like with me?
Oh wow. That's awesome.
Yeah.
So great. That's so
There you go.
interesting. Thank you. It was fun.